Hello everyone. Welcome back to Dr. Lou's VCE Biological Channel. Today, let's learn together about the 2022 NHT Biology. We will focus on questions related to responding to antigens and acquiring immunity. My explanations may need to be revised. If any teachers, parents, or classmates find errors, please feel free to provide your guidance. The molecular constituent that is targeted in autoimmunity is called a self-antigen or an autoantigen. By contrast, a molecule from an infecting organism that stimulates an immune response is called a foreign antigen or non-self. Here, receptors on liver cells are self-antigens. Others, such as pathogenic bacteria, malaria protozoa, and pollen are non-self antigens. When T helper cells encounter an antigen-presenting dendritic cell, the antigen-presenting dendritic cell presents the antigens, and T cell receptors receive these antigens. These receptors are complementary in shape to the presented antigen, so A is correct. T helper cells cannot promote the dendritic cell to divide. B is eliminated. The antibodies are produced by B cells, not T cells. So, C is eliminated. T helper cells help activate B cells into becoming antibody-producing plasma cells. But T helper cells can't become plasma cells. So, D is eliminated. The innate immune system is the body's first line of defense against intruders. It responds in the same way to all germs and foreign substances, which is why it is sometimes referred to as the nonspecific immune system. It acts very quickly. For instance, it makes sure that bacteria that have entered the skin through a small wound are detected and destroyed on the spot within a few hours. However, the innate immune system can't always stop germs from spreading. The innate immune system is nonspecific, so it does not produce antibodies, does not react specifically to a single pathogen, and does not remain specialized cells. The innate immune response is a generalized set of defenses against a range of pathogens. Lymph nodes are sites of B cell and T cell activation. Phospho is a peptide-based molecule since it cannot cross the plasma membrane and binds to the Phos receptor on the surface of the cell. The HIV enzyme could deactivate BCL2. If BCL2 is deactivated, the apoptosis pathway will be opened. HIV enzyme would lead to increased release or less inhibiting of cytochrome C activation of caspase 9 or apoptosis occurring. When the scientists are planning their trial of this molecule, 4 requirements should be considered. Use a large number of HIV-positive participants. Have one group, experimental group given the molecule and one group given a placebo as a control group. Measure the effectiveness of the molecule by the percentage change in T helper cell number pre- and post-trial. Have appropriate control variables, for example, same gender, same duration of treatment, same sample volume of thymus tested for T helper cell count. Repeat the trial to confirm the reliability of the results. Describe 2 components of the immune system's first line of defense that the virus overcame to infect Morgan. Mucous membranes make it difficult for viruses to adhere. Nose hairs are a physical barrier to the virus. The acidic environment of the stomach. Sore and inflamed throat due to the destruction of cells in the throat releasing chemicals that cause increased membrane permeability and swelling. More immune cells moving to the site due to vasodilation. Mast cells release histamines, which produce inflammation. The virus is transmitted by disease-containing water droplets that are spread through coughing, sneezing, sharing food, or close contact. Prevention methods include not sharing food, wearing a mask in enclosed areas, and using hand sanitizer. Since Pat had influenza vaccination this season, Pat did not develop symptoms of the flu after Pat's contact with Morgan, who was infectious. After contact with Morgan, Pat's immune response. Viral antigen identified as non-self. BT memory cells, previously produced, become active plasma cells produced. Antibodies rapidly produced or already present. The virus is neutralized or agglutinated and unable to infect cells. In responding to antigens and acquiring immunity, 4 multiple-choice questions are worth 4 marks, and 2 short-answer questions are worth 18. The overall score for this section is 22 marks. With a total possible score of 120 marks, the percentage for this section is 18.3% of the total score.
If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Lou's VCE Physics channel, Dr. Lou's VCE Chemistry channel, and Dr. Lou's VCE Biology channel.